donor-derived human hepatocytes for liver hepatic. Okay.
Thank you very much. Thank you for the invitation to talk today, and to Jesper for showing me what midsummer is like in Sweden. It's very good fun, I'll tell you that. So, as I said, I work with Dave Hay, and the focus of our lab is to derive hepatocytes from pluripotent stem cells. So, the main issue with current cell-based models for modelling disease, toxicity studies, and just to look at liver development is there's many advantages for each cell type, but there's also a huge number of issues. For primary human hepatocytes, they're limited by supply, expense, and have variable quality. Hepatic cell lines can have phenotypic instabilities. Animal-derived hepatocytes have obvious issues, and hepatic progenitors are suitable but are immature. And a lot of these systems don't accurately model human development. So we have chosen to use a different kind of cell. So we use ES and IPS cells, which are derived from embryonic and adult tissues. These can be differentiated into various hepatic cell types and used for a variety of different purposes, for looking at disease-specific cells, disease modelling, for gene targeting, development, transplantation, and looking at normal human development. And all of these can be used for drug discovery. The hepatocytes that we produce in our lab are 90% pure, so they're pretty homologous, and they have nice morphology. They look quite similar to primary hepatocytes. They're also scalable. We can grow them in loads of different formats and shippable. I believe some have arrived here this week. We can also use them to test various different drugs. They contain sugars and can be transfected with viruses, and these cells are also polarized. So this is how we make them. We have a stepwise differentiation protocol where the cells move from pluripotent stem cells through to definitive endoderm, to hepatic progenitors, and then finally to hepatocytes, or hepatocyte-like cells. This process takes approximately 18 to 20 days and involves 3 different media changes and can be done on a variety of substrates. However, for Matrigel, which is a fantastic product, it has its downfalls. The cells at day 17 to day 21, we have nice hepatocytes, nice morphology. You can see here nice defined outline of the cells, and they have high cytochrome activity. So cytochromes are a key enzyme involved in liver metabolism, and this is highest at day 17 and then decreases gradually. This is due to the phenotypic instability. As the cells age and the Matrigel ages, the cells lose their morphology and lose production of transthyretin and CYPs. And as you can see at the end, they look pretty miserable. So in an attempt to overcome this, we looked at screening various different polymers. So this is in collaboration with Professor Mark Bradley at Edinburgh University, who developed this microarray approach for using 384 well plates, and then we can print triplicates or replicates of the polymer onto agarose slides and then culture cells with these to screen high throughput to see what our cells would like. So we took our hepatoblasts at day 8, 9, 10. These were then plated on top of the slides containing the polymers. They were incubated for 8 to 15 days, and then they could be fixed and stained and then imaged. This helped us narrow down from hundreds of polymers down to about 6. So we got 3, so the first one here is pluripotent ES cells, and then we have cells on Matrigel. We have 3 polyacrylates and 3 polyurethanes. This is freshly preserved primary human hepatocytes. So we're seeing two key proteins, fibrinogen and transthyretin. And a key, our hit polymer was polyurethane 134, which drastically improved the cell function. We also saw that on this polymer, 20 days post-replating, that the cells expressed much lower levels of the dedifferentiation marker vimentin, and that HNF4 alpha, a key transcription factor, was maintained. We also then went to take this polymer and we used it to screen various iPS cells and ES cells to determine their functional activity on this. As you can see here, both iPS and ES cells were highly expressing cytochrome 3A and 1A2. So now we had a model that we could use. We went and took this to Bristol Myers Squibb, a drug company in America. We then identified two compounds that could be metabolized specifically by CYP2C9 and CYP2D6, and then we used these to screen for how functional our cells were. So when these drugs are metabolized, they're broken down and they become toxic to the cells. So toxicity is a marker of how functionally active these cells are. So we can see here that the second compound, we got high toxicity in both iPS and ES cells, but the first compound, BMS1, did not induce toxicity in iPS cells. This is due to the fact that these iPS cells do not have the cytochrome 2C9, so this shows that our system is very specific, and we can also use iPS cells with certain metabolic defects to analyze drug metabolism. But we then came across another problem in our culture system, was using serum. We saw huge batch-to-batch variability in cytochrome activity, and we then tried to eliminate this as an issue in our system by moving to a serum-free manufacture. So you can see at day 18 we've got some nice looking cells, and these progress in a stepwise way through losing their pluripotency associated genes, increasing endoderm and early hepatic genes, and increasing cytochrome gene expression as well. This was reflected in protein production and through flow cytometry analysis. This was highly efficient, with approximately 84% of cells expressing FOXA1, 94% in HNF4 alpha, high expression of AFP, and then finally the more mature albumin expression, we're seeing 97%. This process can be translated to GMP grade cell lines. As you can see here, the AFP production is alpha-fetoprotein. It's an early hepatic marker. So we see slightly higher levels of this in our MAN11 cell line, and significantly higher levels of albumin in this cell line. Again, with different batch-to-batch variability or different cell lines, we experience different cytochrome activities as well. So now we had a good system. We used this to scale up and to look at various different drug metabolisms. This looks incredibly complicated, but all it's basically saying is that on day 7, our cells predicted toxicity to similar levels of cryoplateable hepatocytes. So they're sensitive cells. They know what they're doing. These cells could also be infected with hepatitis C, which affects 2 to 3% of the world's population, and can lead to severe liver disease and liver cancer. So our cells express viral entry factors, including CD18, SRB1, and Claudin. But when compared to the hepatocarcinoma cell line Q7, there was much lower levels of RNA. This is because the cells were not infected at the same rate that this cell line was. This is due to the fact that our cells have innate immunity, We then went on to look at what kind of immunity this was, and it was a type 3 interferon response, as identified by key interleukin genes. We then inhibited this interferon response and showed that we got increased transfection or infection of our cells. So this is all stuff produced by other people in our lab. I'm going to go talk about what I do now. So that's the tricky stuff over, hopefully. So as I said before, we can make nice looking hepatocytes. They have high levels of albumin, and they express it nicely. They have high levels of ecadherin and high CYP function. But we have this issue that the cells are not mature. So they're producing high levels of an immature protein, and this is a common problem with this kind of production of hepatocytes. So we then looked at different ways to overcome this, and one of them is to use a tissue engineering response, so to engineer the cells in 3D. So we took our hepatocytes at the progenitor stage, when they're more amenable to lift up and to be encapsulated. These cells at this stage can produce cholangiocytes and hepatocytes, and they can be replated over several passages without any significant change in gene expression. We then encapsulated these cells in alginate beads, and we saw a dramatic increase in albumin production, and more importantly, a significant decrease in AFP production. However, 20 days later, the majority of cells were all dead, so this obviously needs improvement. So we then went on to try and find various things we could improve this system with, by looking at various synthetic and biological factors. So we went back to our polymer screen where we saw similar polyurethanes, 9G7, combined with different hydrogels produced really nice high levels of cytochrome activity, but were incredibly variable. This is due to solvents and the way that the polymers are manufactured. It makes them very tricky to work with. However, so then we went on to look at more biological substrates, and here are 2 laminins. I apologise, I've been told by my boss to take out which laminins is until this is all published, hopefully. So we're going to call them A and B for now. So here, laminin A and laminin B, we saw really nice metabolic activity, and more importantly, it was much more reproducible. So laminins actually support hepatocyte gene synthesis in a similar fashion to Matrigel, using the same media, the same culture conditions, but just culturing the cells on laminins from day 1 or day 0. So day 0, they express pluripotency associated genes on Matrigel, which is the black. The checkered is laminin A, and the grey is laminin B. So we see a nice decrease in pluripotency associated gene expression, a really nice upregulation of the immature marker AFP, significantly higher on laminin A, and we saw a similar trend for HNF4 here, so at day 9 when they're expected to produce immature markers. But then this is where it got really interesting. We saw significant increase on the laminins for albumin production, the more mature gene. So we then went on to see how many cells are expressing these markers. So at the hepatoblast stage, so the progenitor stage, we saw high levels of, this is AFP in red, and HNF4 in green. So we saw comparable levels across all 3 substrates, really nice SOX9 expression and CK19 expression. You can see how here the cells in laminins were much larger, and some of them were starting to become binucleate compared to Matrigel. We then also saw that as we moved later on, that the cells were producing similar levels of albumin at the protein level, and what we thought was quite interesting was that the cells were proliferating far less on the laminins compared to the Matrigel. So they're maybe being told to slow down, differentiate, don't proliferate. As I said before, we saw really nice hepatocyte function. This was increased So each one of these is a significant increase apart from where it says NS. So instead of dazzling you with too many stars, I've just said where it's not significant. So both laminins, we saw huge improvement of our cytochromes, cytochromal activity, and you can see really nice immunostaining here as well. So I'm sorry, I've flown through that way too fast, but there we go. Sorry. So in summary, our laminins can be used as an alternative culture substrate to Matrigel. Hepatocyte-like gene expression is supported on 2 types of laminin, and the morphology of our hepatocyte-like cells is enhanced. The metabolic function of the cells is significantly enhanced, and the protocol is now GMP translatable. So thank you all for paying attention, and to everyone that's been involved. And I've got a poster tomorrow, so
Great.
thank you all.